All right, thanks for the invitation to join the meeting. If you could see that picture that Nikolai keeps showing, you can see that I'm in the background on one of the pictures. At the time, I wasn't doing anything to do with single cell proteomics, but I was very curious about it because and what people were doing, because I kept getting asked by program officers at NIH, what's the status of single cell proteomics? So I figured there must be a lot of interest in what's going on with this. So most of the work, a lot of the work that we do in the lab tends to be centered around neurobiology, and so it kind of made sense for us to try to integrate single cell with neuron analysis and some of the other studies that we were doing. And I actually had somebody coming from China who was going to spend a year in the lab, was going to work on this. And then when the doors got shut because of the pandemic, that was delayed. And these were things that we were going to do with Stuart Lipton's lab, and that they all resulted from a hallway conversation about wanting to do single cell proteomics, and he was working on some Alzheimer's disease neurons using iPSCs. So we decided to get started on this, and Jolene Diedrich's been doing most of the work together with Matt and the other people in Stuart's lab that do patch clamping and so forth. Okay, it never likes that arrow. So one of the things that's really interesting about neurons, I think, is that thought and critical thinking is uniquely human. And of course, that's the arrogant human perspective on this. And most of us that have been spending time during the pandemic at home with our pets know that we're easily manipulated by our pets. And so to think that they're not using critical thinking to manipulate us is a little arrogant, I think, on part of that. But what is interesting is that, you know, neurons, we're learning that neurons have particular circuits that are dedicated to particular sorts of activities. And it's not, you know, massive numbers of neurons that are involved in this, but it's much smaller numbers of neurons than we previously appreciated. And a lot of that's being learned using the techniques of optogenetics as a way to tease out how these neuron circuits are being used to control certain types of behaviors. And what's interesting, of course, is that bigger brains result in smarter animals, more neurons with more synapses, but this, of course, is not the whole story. And you can clearly see that there are differences between the human brain and say a cat's brain. And so the human brain is much bigger, and there are many more folds in it. And the folds are one way that evolution has tried to pack more capacity into the brain, which is ultimately, the size of the brain is ultimately limited by the ability of females to pass infants or fetuses through their to give birth. And if the brain gets too big, then both the mother and the child die in childbirth, and that doesn't work well for evolution. So the workaround for that was to create more surface area by increasing the folds. But total neurons, so size results in more total neurons. And if you look at the difference between cat and humans, we've got about 85 times more total neurons in the system than they do, but we only have about an order of magnitude or more total synapses than do cats. And I'm sure you've all been watching cute cat videos on YouTube this entire pandemic, and you know that cats are actually quite clever and do interesting things, given the size of the brain and so forth. So what's interesting is that, of course, is that it's very difficult to study the evolution of thought and brain and brain power and so forth, because it's a soft tissue, and of course it doesn't survive the death process like solid materials like bone do. And so all we really have had to assess the capacity of human brains over time is really the size of the cranial cavity. But one of the other things that people look at is to look at the evolution of thought and critical thinking and intelligence and so forth. is what humans do with their hands and their brains in terms of thinking about things. And 300,000, 500,000 years ago, this is the sort of art that humans were creating, and then they started to get better at doing this and more imaginative. This clearly is not just an exact reproduction of something they had seen, but they were using parts of their imagination to try to figure out, to make this sort of character. And that's about 35,000 years ago. And then you can also see this other structure, which has a little more detail in it than the other one. And then, of course, 500 years ago, Michelangelo was carving David. I assume this is a exact reproduction of someone. May have been me, 30 years ago, but certainly not now. And then, of course, Jackson Pollock was creating this sort of art fairly recently in time. And so you could consider this as I guess, as a step forward, or you could consider Jackson Pollock's art as a step back in time, although this involves, I guess, much more abstract thinking. And of course, humans were building things. So almost 700 years ago, the Notre Dame Cathedral was completed, and took about almost 200 years to build it. And I assume there's a lot of trial and error that went into that process to build this. And nowadays, we're building things like that are heavier than air, that can fly, and that are just magnificent feats of engineering, such as CERN and the A380. So primary challenge with studying the human brain, of course, is that it's difficult, if not impossible, to study human brains at the molecular level in situ. It's very difficult to convince somebody that you want to go in and biopsy their brain and take it out and study it, unless there's some sort of surgery that's required in order to save their lives. But most studies of live human brains use non-invasive spectroscopic techniques like functional MRI or MRI and things like that. And in model organisms like the mouse, we can now do optogenetics, which allows interrogation of brain circuitry. But of course, you have to create a transgenic mouse that's expressing in a neuron-specific manner certain ion channels that allow that are sensitive to light wavelengths that allow you to activate those circuits to be able to test the functions of those particular circuits. What's been interesting over the last decade, I guess, is human brain organoids have emerged, and these will allow the study of disease genotypes and possibly function. It's just not yet clear how well they will be recapitulated in organoids. And we've started doing some work in this area, not at the single cell level yet, but we've been working on these things to study particular types of disease genotypes, such as Rett syndrome, to try to sort out what sort of problems are going on with neurons. So single cell molecular analysis in neurons is not a new thing. In fact, it has a fairly long history, being led by Sweedler and a lot of the offspring from his lab that have looked at model neurons, such as Aplasia californica. But they've also extended the techniques that they've developed in these particular neurons into different types of cells. And Sweedler, he actually started this work and very intentionally did a postdoc to learn how to use Aplasia californica to do these studies. And he's been doing this for, I guess, almost 30 years now. And Aplasia is a good model system to study individual neurons, primarily because the neurons are large. And so he's used a variety of techniques, MALDI, capillary electrophoresis, and mass spectrometry to do this. And he's also measured a variety of molecules. But he hasn't done a lot at the proteome level in these single neurons, primarily because it didn't until, I think, fairly recently have a lot of genome information associated with it. So he's mostly looked at neurotransmitters and neuropeptides, which are much easier to interpret on smaller scale. But the important point is that Sweedler established the importance of single neuron analysis at the molecular level, and has been doing so over a number of years. So it's been rather interesting. And I think what I think, after having been around science for a long time, what is interesting is that people can be laboring away on certain things for a while, and then all of a sudden they become hot and everybody starts taking notice of them. And I think Nikolai and Bogdan certainly get a lot of credit for having brought the single cell analysis, proteomic analysis to the forefront by looking at smaller cells, which are much more interest than some of the larger cells like neurons. And so this is some of the work that Sweedler's lab has done, where he's looked at individual organelles with mass spectrometry, and he's tried to characterize feeding circuit behavior in Aplasia, primarily using a lot of MALDI techniques, because you can do these things in a spatial manner. And some of his offspring, Lingjung Li, for example, has been also using mass spectrometry, in particular MALDI, to look at particular kinds of circuits in crustaceans. In this particular paper, in this example, she's been doing it in crab. I remember seeing one of her proposals many years ago come across the study section where she was trying to look at feeding behavior in lobsters, and I was very suspicious of that grant proposal. You know, what are you going to do with the lobsters when you're done with these studies? You know, are you going to sacrifice them in a humane way? And I'm sure they wound up in a boiling pot of water and as dinner. So the efforts that we've been doing with Lipton's lab are to look at Alzheimer's disease. This is, everybody's familiar with this. This is a chronic neurodegenerative disease. It affects about 5.8 million Americans, about 50 million worldwide, with about 10 million new cases each year. It's a side effect, I guess, or a consequence of longer lifetimes that result from improvements in medicine. So in 2015, the total global societal cost was estimated to be about $818 billion. Results in memory loss, problem with language, disorientation, mood swings, loss of motivation, problems with self-care, behavioral issues, dementia. This all sounds like the symptoms from stay-at-home during a pandemic to me. This disease process is also associated with plaques and neurofibril tangles in the brain, although some of the problems associated with Alzheimer's disease occur much sooner than these other obvious symptoms of Alzheimer's start appearing. So plaques and neurofibrils and so forth, before they really start becoming noticeable in imaging techniques and so forth, start experiencing memory loss. So this is a massive failure in proteostasis. So these proteins that start to misfold, they're not managed very well for various reasons. The genes that are associated with familial AD are quite well understood, but sporadic AD is still a mystery in terms of what is driving that particular disorder. So one of the issues seems to be a vicious cycle of neuronal hyperactivation. This is what Stuart's lab has been starting to study. So this is the plaques that are formed, AB aggregation, and so forth. They preferentially activate neurons by blocking glutamate reuptake by neurons and astrocytes. This leads to hyperactivation and eventual neurodegeneration. And so the glutamate reuptake by the neurons gets slowed down because it's blocked by the plaques that are starting to be formed. And so this results in hyperactivation. And so the approach that Stuart's lab has been taking is to use human-induced pluripotent stem cells. And normally you would take these from somebody with the disease genotype and do this. And this is what we've been doing with Rett syndrome and other disorders, where we're going to start using the same approach to study those. But what Stuart's lab did is he took the AD neurons and generated a methionine to valine mutation to position 146 and the presenilin 1 gene of wild type skin cells, and used CRISPR-Cas as a way to make that change. They then reprogrammed to the embryonic state. They created human iPSCs, differentiated neurons by dual SMAD inhibition protocol to form neuroprogenitor cells, NPCs, that are subsequently treated with BDNF and GDNF neurotrophic factors. And then these are plated with mouse astrocytes and they undergo neuronal maturation. And after about 4 weeks, mature neurons are ready for experiments. And this is a rather common procedure that's being done now as a way to study neurons with particular disease and genotypes. Now, these are neurons like to be in communities. These usually result in two-dimensional cultures, but one of the things that happens when you make the organoids is that you're able to create these basically three-dimensional cultures, and they start organizing and forming morphology that's similar to what you would see in the brain. So the patch clamp technique was developed a number of years ago, and just interestingly on Twitter, there's been this discussion about electrospray, and this guy Nima, who won the Nobel Prize for using patch clamping techniques in live cells to study ion channels as part of his master's degree when he was at the University of Wisconsin on a Fulbright scholarship, developed an electrospray source in 1967, so before Dole, before Fen, and was spraying large molecules and viruses and things like that into a molecular beam instrument. And I guess he never published it because it only existed in his master's thesis. But he then went on to do patch clamping and won a Nobel Prize for that. And the patch clamping is a very common technique in neurobiology. It's a way to measure electrophysiology in neurons. And it's quite commonly done. And you can see in this picture over here, this is a picture of a neuron being patch clamped by a capillary. So all those pullers that we all have in our lab to make electrospray tips, those are designed to make these sort of tips for doing patch clamping techniques. So you're set up with basically a glass electrode, and you are measuring currents in these cells. And this is some patch clamping that's being done in wild type neurons versus these AD neurons that Stuart's lab has created. And so in a normal wild type, you would see this action potential, to see sort of random firing of the neurons, but in the AD neurons, what you see is very hyper activation of the neurons. You can also measure capacitance using these techniques, and what that tells you is how many synapses are available, or how many synapses are formed on that particular neuron. And so you can see this bar graph here that there is quite a lot more spontaneous action potential occurring in the AD neuron than in the wild type neuron. And the capacitance in the AD neuron is much lower than it is in the wild type, suggesting that there are fewer synapses that have been formed. AD neurons also manifest shorter neurites than wild type neurons. So in these immunohistochemistry stainings, you can see the MAP2 protein is a marker of dendrites. And then the green is the TUJ1, which is a marker of pan-neuronal marker. And so what you can see in these measurements is that the neurites are much shorter in the AD neurons than they are in the wild type neurons. So in order to pick out neurons that are meeting all the requirements for showing AD phenotypes, a number of different measurements are being made by patch clamp. So there's the RMT, which is the, what does that stand for? I forget. And then there's sodium density, potassium density, capacitance, and then spontaneous action potential. So this has to do with root mean potential. So it's just what's happening at that steady state. And this can be measured with the patch clamp. So the ideas that we had developed with this was to go in, Stuart's lab would go in, they would measure, make sure that they have, make all these measurements on electrophysiology to make sure that they have adequate AD neurons present in the patch clamp. And then what we would do is apply a small negative pressure onto the patch clamp electrode and suck up the neuron into the patch clamp electrode. We then expel the contents out, digest with trypsin, and then put it directly into the mass spectrometer. So fairly simple workflow, so nothing particularly elaborate here. And unlike a lot of the work that we've already seen, particularly the work from the Geiger lab and so forth, where they're looking at cancer cells, which are much, much smaller cells, and the cells that Nikolai and Bogdan are looking at are much, much smaller cells. The neurons are even human neurons are much bigger cells, and so there's much more protein present in there, and which, while it's still a challenging analysis, it's certainly not. It's not like looking at tiny cancer cells that are proliferating in pancreas or something. So of course, the first attempts at this, we didn't see much, and so they made some adjustments to the protocols. And one of the adjustments that we made was to digest with trypsin 60 degrees, 1 hour, and then go directly into the mass spectrometer. Now, and out of that, we actually got some decent data. And what that tells me is that, so it was a rather simple adjustment to the protocol in order to get to that point. And what that tells me is that with some additional adjustments, we should be able to improve our data acquisition quite a bit. And then went into an Eclipse mass spectrometer, 140-minute gradient using an easy NLC. And the data that we've collected, we did this on about 20 AD neurons and 24 wild type neurons, which passed screening protocols and were collected for mass spectrometry. And so we are now at that point where we're we're starting to replicate this this first analysis to make sure that we get the same data. So here's the proteomics pipeline for our single cell analysis. This is an Orbitrap Eclipse mass spectrometer. I'm sure you're all familiar with that. And then we do our data analysis. So our MS files are extracted with a raw converter. That's just a converter that we wrote for converting thermo data into flat files to be searched. We then search with our search program called Prolucid. This is like third or fourth generation sequest against the Uniport human single entry protein database. And then we use a program that was written by Paulo Carvalho called Pattern Lab. Pattern Lab was, he wrote this when he was in my lab many years ago he was doing the international part of his PhD program. So search labs are filtered and compared with Pattern Lab, usually in NSAF for comparisons through this. We're also continuing to do some additional data analysis with the data that we've collected to see how much we can improve on it. So these are the results that we have. These are the analysis for the peptides. So this is the data that we collected. This is 20 AD, 24 wild type runs. We had one failed MS run. After filtering out the data, we have the data we collected or kept was for 14 AD cells, 18 wild type cells, about a 70% success rate. We collected information on about 700 proteins from about 2,000 peptides identified per single cell. There are differences between the wild type and the AD. At the peptide protein level, we're seeing about 81 that are unique to AD and about 269 that are unique to wild type and about 624 that overlap. At the peptide level, we're seeing about 1,500 that are unique to wild type and 186 that are unique to the AD set. So these are identified in at least one sample of proteins and peptides that are identified in at least one samples. But for protein identification, we required a minimum of 3 samples per genotype in order to keep the protein as present. Again, as I mentioned, we're still working with our data analysis here and trying to get a feel for this. So this is differential protein expression in the wild type versus AD single cells. And we are seeing things which are what we call positive controls for what we might expect to see in an AD sample. So the proteins must be identified in three samples, single cell. The red 843 proteins do not satisfy fold change or p-value. So these are the ones that are not changing enough. The green 82 proteins satisfy fold change, but non-significant p-values. And then we have blue, about 32 proteins, and yellow, 42 proteins, both satisfy fold change and significant p-value. Yellow are low abundant proteins. And so we're seeing things which are clearly shown up. This is our MAP2 marker, which is the immunohistochemistry marker for dendrites. And we're seeing that as being significantly changed in these samples. So we take this through some correlation analysis, heat maps differential protein expression in wild type versus AD. We also did PCA analysis to see if we could separate the AD proteins from the wild type proteins. This is a poor man's UMAP. We don't really have enough data to be doing UMAP at this point. But the partial least squares loading plot, this is AD versus wild type samples. It's used to determine proteins that contribute to differences between the cell types. And I'm sure you can't read this because I can't read this on my screen either, but we're seeing that proteins are clustering between on the side of AD and on the side of the wild type proteins. So some proteins detected as differentially regulated in this single cell analysis have also previously been implicated in AD pathology. So these are our positive controls. Cathepsin B is increased in AD relative to wild type. We also see metalloprotease inhibitor 2, so TIM2 is increased in AD relative to wild type. The function of TIM2 is a tissue inhibitor of metalloproteinase 2. It is known to be elevated in AD patient CSF. Alpha-tubulin N-acetyltransferase is decreased in AD relative to wild type, and it functions to acetylate microtubules promote proper bidirectional axonal vesicular transport. And we're seeing differences in that as well. MAP1B is decreased in AD relative to the wild type. So we're seeing that change. Drabrin is decreased in AD relative to wild type. We're seeing that change. And neuronal migration protein double cortin DCX is decreased in AD relative to wild type as well. So we're also seeing that change. We're also seeing dynin and dynactin is decreased in AD relative to wild type, and the function of dynin is the primary ATP hydrolyzing motor responsible for retrograde axonal transport along microtubules, and works as a dynin-dynactin complex. And so it's some suggestion that this may be critical in Alzheimer's disease. So conclusions in our further work. So we successfully implemented patch clamp LC-MS on single neurons. While time consuming and low throughput, the patch clamp screening for neurons prior to selection for MS analysis is essential in obtaining high quality neurons and electrophysiology behave as expected. Options to increase throughput are to implement the Slavov-Budnik method with TMT that will require some innovative sample manipulation protocols on our side as a way to figure out how to do that. In other words, minimal manipulation of sample only was key in providing high quality MS data. We identify about 700 proteins per single cell. We think we can increase that. We haven't done any of the tricks that people have used before, like match between runs and things like that. So we're trying to figure out some of that, and we can also create libraries from the data that we've collected and use that as a way to try to improve on our analysis, even though this is DDA data. Certainly we will, at some point in the future, look at DIA data as well. So we've analyzed 20 to 24 single cells per genotype, and we were able to detect differentially regulated proteins that have been shown to be modulated similarly in the AD literature. And so that's a good first step for us. These experiments were primarily proof of concept, try to figure out if we could actually do this and make it work, and then see data that seems to be relevant to the actual disease state. We have observed several differentially regulated proteins in the single cell MS data that are highly correlated to decreased neurite length that we observe in our previous immunohistochemistry data, and could lead to lower capacitance values that are measured by patch clamp in AD versus wild type. And current data contains measurements from both excitatory and inhibitory neurons, and further categorization of the proteomics data based on electrophysiology parameters could lead to additional information. I think one of the really interesting aspects of these studies is the fact that we're able to look at the electrophysiology and try to connect it to molecular data that we see in the cells. One of the things that I really would like to get out, I've always had a keen interest in being able to look at membrane proteins, because they're always so hard, and nowhere, probably no other cell type is membrane proteins more critical than they are in neurons, because that's where all the action is, particularly at the synapses and so forth. And so these are people that were involved in the work. So this Jolene Diedrich was doing all the mass spec in my lab, and she was involved in developing the protocols for doing the mass spectrometry. Salvador was involved in informatics. Antonio was helping Jolene on some of the mass spec, and Matt and I'm not sure who was doing the patch clamping. It may have been Nima or Swagata or Maria, but these are people in Stuart's lab that have been involved in this particular project. And thanks.